Hello, everyone. My name is Xin Lei, and I am a senior research investigator at EMS in clinical pharmacology and pharmacometrics. And I am glad to serve as a moderator for this panel one discussion.
Hi. Hello, everyone. My name is Wei Xiong. I'm come from Melteni Biotech. Right now is the project lead in the translational medicine. Yeah, thank you all the attendees. Okay, let's see. Yeah, I think everyone like me is very exciting about the presentation in this session. Thanks all the speaker do this outstanding and exciting presentation. We have heard a fantastic the range of perspective from AI-driven the drug design and small molecular therapeutics and cardiac metabolism. Welcome all three the speaker join us for this panel discussion. So let's see how the audience do have any the question to the speaker. Yeah.
Good morning, everyone. Good morning, speakers. My name is Sia. I come from Johns Hopkins Medicine, and thanks very much for speakers' outstanding presentation. So I have a question for Dr. Su, and your presentation is really impressive for your NAP introduction, the disease, and the research and summary. And thanks you very much. And since my own background from pharmaceutical science and now medicine, so I'm a little curious about for the first question is why you changed from your medicine pathway to pharmaceutical science, because as a medicine, the thinking way, usually we only use the top to 200, 300 drugs, and usually providers use only their preference list drugs. So what's the reason drive you to change your pathway from medicine to pharmaceutical science? The second question is, so I know you, your department, your team put a lot of energy and very excellent team members, but compared to the pharmaceutical science companies, it's a little too much energy and too many people put in. So I really want to know what's your final goal. I know you already moved a long way and achievement, but if you want to achieve final drugs, yeah, what's your final goal? No offensive, no offensive. If you feel uncomfortable, I still feel grateful. Thank you very much.
Well, first, my career. So the short answer is basically, so we have two body issues. My wife, she's a physician, and we just can't handle, two doctors in the same house. So, and also, so my, a little bit, a few years ahead of my wife. So when she's preparing board tests, so I'm already launched a job. So, and also when I completed my medical school, so I really want to do some research. And I would like to pursue a career that can bridge my medical knowledge and also my research interest. So then, so at that time, so I chose to do clinical pharmacology. So the reason I came here in the United States is because I was funded by Merck Foundation as a clinical pharmacology fellow. So I did my clinical pharmacology fellow in UCSF. And then, so my mentor, so she suggested, we got a great opportunity. So if you would like to pursue a PhD here, so you're welcome. So that's why I did my PhD at UCSF. And then so it looks like it's very logical for me to pursue my research interest in pharmaceutical science, because my PhD is a pharmaceutical science department in pharmacogenomics. So that's the first question. So the reason I choose the pharmaceutical science research field. The second question is yes. So my department, so we have different faculties. We're doing from very beginning basic science to the clinical research. So yes, compared to those pharmaceutical companies, we're small, still small. Even we have not a small department, but I think so there are advantages versus disadvantages. So the advantages is we're small, so we can make decisions relatively easily. I know folks here, they probably have so many meetings. So we don't have many. So we can, for example, I just talked to my collaborator, can you, redesign this function group? And we would think, it's probably important. And then the next day, so he can start it, right? So that's the advantage. So the disadvantage, of course, we don't have so much money like those guys have. They can have different teams to support their work. And I think it's eventually. So we would like at the time, for example, our compounds now. So if we really got a very safety compound, probably we need to look at those investors. Probably talk to folks here or folks in other pharmaceutical industry. So I think did I answer your question?
Thank you.
Thank you, Dr. Shu, for this
okay. That's next.
answer. And yeah, please go ahead.
Yeah, thank you very much. I first just like to compliment all the speakers. I've enjoyed it very much. But my question is for Dr. Wang. As you touched briefly the democratization of AI, and I could testify firsthand because I'm not very good at it, but we've been doing using Boltz-2 and docking peptides and small molecules. And for the people in the audience, I would recommend all of you try Boltz-2. It's really a wonderful program, and they just launched Boltz-Gen from MIT. But my question is, what I noticed is that we did not have a crystal structure. We built our model with AlphaFold 3, and perhaps that is the reason. But I think even when you talked a lot about crystal structures, the crystals are not perfect in terms of the true structure. And I think what's missing in the field that perhaps you could contribute is that I think biochemistry trumps these modeling. And so we've docked peptides and molecules, and we find disconnects between the model prediction and the actual raw data. And so I think it's an iterative process that has to undergo. And I think people like you or people in the community, we need to build tools that allowed amateurs like myself to kind of build upon what we're finding, because the real data trumps models, but there's not that connection yet. So I guess my general question is, could you just talk about crystal structures versus cryo-EM versus models, and how good they are in predicting docking and identifying small molecule targets?
It's a very good question. Yeah, I appreciate it. So I think before the AI age, we have so many tools. We have homology modeling or comparative modeling. For that case, we can find the template close to your target, right? So, but now in AI, I say since the AlphaFold came out, now we have now the latest version, AlphaFold 3. You can really try that. So the AlphaFold and Rosetta and AlphaFold, they win the Nobel Prize last year, right? So yeah, try both. So the accuracy of AlphaFold 3 is very high. It's very high. So also, I mean, it depends on your target, right? So if your target already have a lot of structure available, So I'll say that in that case, the accuracy it could be guaranteed because it's in the training set. But if your target is not that well defined, so or maybe have some like loop issue, right? So yeah, I mean, just like you said, so you actually you can fine tuning your models. You don't have to just use that directly. I mean, it's really recommend you do that in the iterative ways, right? So yeah, if you have the more data, you can put in the large model and you can fine tuning, right? It do much better. But I mean, is really recommend you go that way. Yeah. Okay.
I have two questions. One for a short one for Dr. Xu. So for the molecule that you and your partner optimized, right, from a low bioavailability to a high one. So a short question is whether AI play any role in the optimization.
Not yet. So that's we had this collaboration actually several years. I think the AI, so we began to do the AI starting actually from this year. So in this particular, those particular data I talked about today, so we don't have any AI component yet.
Yeah, then I think future you have a lot more candidates with AI assistance. The second question is to Dr. Wang. And so as those drug discovery companies are switching human labor with those chips, GPUs, I wonder whether you can give all the people who are involved in this industry some general advice, especially for the young people still developing their career with this current tide of switching from warm bodies to the GPU.
Well, I'll say, yeah, yeah, I really encourage just to use it. Actually, don't get scared of this kind of like the big program, right? Like AlphaFold 3 or ResettaFold. Yeah, so actually they are very easily accessible. So most of them they're open sourced, or you can get access from the Google Colab. Or of course, you can use all the cloud computing like AWS, or I mean, if you don't want to buy the CPU, so yeah, you can use in the cloud computing, but I'll say so yeah, just I mean I'll say that now most of this very good famous program, and you don't need to buy that, right? It's all most of them are open sourced. I mean, why not just use it and right? I mean, you can get it like right get some experience, or you can hire some people, right? Just like you can learn from this kind of like this process, yeah. So it can really, I'll say, it's really powerful. It's really powerful. It can really solve a lot of problems, and also if there's no copyright issue, I mean, so you can use in the public, like the cloud computing platforms. Yeah. But if it's okay.
Thank you, everyone, for the very wonderful talks you give us. So I do have a quick question for Dr. Wang, Professor Wang. I noticed that you showed a lot of data from your group. I think that's very exciting, especially like the first, sorry, Dr. Su Yan, yeah. So you mentioned paravalent and also YA6060. I think the data are very exciting. As an oncologist, I do have a question for you about the oncology side, because I saw you show the data from colorectal cancer, because we were trying to connect or link the metabolism and the cancer, right? So I understand, like you also mentioned about. So I wonder whether you have any data from liver cancer. Another thing, since we're talking about metabolism, so I'm thinking about insulin. Is there any data from pancreatic cancer? We know that pancreatic cancer actually in the oncology field is a very hard one. So I wonder whether there's any new signal from your data. Thank you.
Thank you. So we do actually have lots of data for cancer, but we are still focused on colon cancer. the reason I actually mentioned is because colon cancer, more than 90% of patients, their tumor samples, they have upregulated Wnt signaling pathway. It's very straightforward to look at colon cancer first. But we have a plan to extend our study to our compound to other cancer indications as well. The reason is Wnt signaling is a fundamental pathway that play a role in a variety of cancer types. And also, like you mentioned, the metabolism is also fundamentally changed in cancer tumor tissues. So in particular, for example, the AMPK pathway, so there very strong data suggest the AMPK activity got suppressed in cancer tissues. So yeah, we do have plan, but right now, so we don't have any data for pancreatic cancer, other cancer types of breast cancer. But we do think, our compound probably would be able to have effect on cancer stem cells because of the metabolism modulation and also the Wnt signaling inhibition. Thank you.
Okay. Yeah, thank you very much for all the speakers' wonderful talk. I have a quick question for Dr. Ruya Liu. So you mentioned it's a hypoxia pathway or HIPPO pathway regulation can change the cardiomyocyte proliferation. How does the change of hypoxia or HIPPO really affect the metabolic profile of cardiomyocyte?
So as mentioned, I probably forgot to mention more, but in zebrafish, initially they found that for the cells to proliferate, they have to de-differentiate to a younger state and then re-differentiate into a more stronger state. So it actually happens later, more articles, more research, they found that it also happens in the mouse. So they haven't, I think, in human, they haven't done this research, but they also found whatever that are proliferating. So anyhow, the background is at baseline in human hearts, there's always 1% cardiomyocytes that are proliferating. Right now, when you're sitting, there's still 1% of your cells are working. And then during MI, there might be an increase of proliferation, and also very recent study that was published on Circulation, I found that even in heart failure that got LVAD, left ventricle assistant device, they would also be able to induce proliferation by 9-folds. But anyhow, the point is, whatever that's happening, all these cells, when they need to proliferate, they have to go back to younger stage and then be stronger. So that's,
I see. So if there's
there's always going back to glycolysis, and then after they're stronger, they can do oxphos.
So potentially there could be a metabolic regulator or drug that helps to the heart to regenerate. In the future.
Yes, actually, there are people doing it that way. I did not mention one of the very important paper that was published on Nature in 2023. They did the other way. They actually blocked CPT1. The carnal. I forgot the name, but they blocked that and then found that there's more cardiomyocyte proliferation. So that's kind of a link of these two pathway, metabolism and proliferation.
Yeah. So my comments, in the future, there might be some drug that can increase the cardioplasticity. So the patient who have for example, a stroke or heart disease, their heart can, recover better, maybe, with the help of the medicine or its metabolic treatment.
Yeah, for sure.
So, yeah, that's great.
Yeah, I have a question. Regarding the facts, during the pandemic, COVID, a lot of people with metabolic disorder and people old age had more serious symptoms. Also, they die. Also in China, even nowadays, a lot of young people after the pandemic, after receive this low quality vaccine, they die at very young age. So the question maybe for Dr. Liu and all three. So what might happen to the metabolism, especially to the heart tissue of these people after injection, vaccine or after COVID? I think 10% people have long COVID symptoms. I think many of people who have affected by COVID still have maybe some residual effects, and it may affect the aging process. See, in China, a lot of people, young people nowadays die for no reason, just quickly pass away. People believe that's due to the COVID residue. So what might happen to the people's physiology, and what's the possible solution? Any drugs? Your three expert.
So you asked about heart first, right? So I'm just like quickly thinking what are possible. So I have not checked the most common cause of death after COVID vaccine, but I could imagine if they happen in younger patients, younger people, then I could think of very likely it's due to arrhythmia, right? Because if a person who's young and then they die, most likely it's just maybe AV block, meaning atrioventricular block. Meaning atria cannot conduct with the ventricle, and then boom, the person died, right? But in terms of chronic effect on metabolism, I'm not sure how much that's gonna impact the metabolism itself. It might be what I could think of, maybe, if at all, anything that's impacting the conduction pathway that's causing the pacemaker cells to not function. Maybe there's more fibrosis happening in the heart. I'm just taking an educated guess here, so don't take any of my words. You can search later online if whatever I'm saying is right, but I'm just making hypothesis here. Maybe some of the pacemaker cells, they got fibrosis, and then that's causing fatal arrhythmia, and then that's causing increased cause of death in younger patients. In terms of how it's impacting the fibrosis, we can also call this impact of remodeling, because may or may not be the cardiomyocyte itself. It could be, the stromal cells that's just kind of intervene among the cardiomyocytes, and how these are impacted by the vaccine. Maybe that's the immune response, because currently the biggest discussion is we're seeing a lot of autoimmune diseases right now in China, right? We see lots of Hashimoto, we see more autoimmune diabetes, and something more. But we see celiac disease. We see a lot more of these happening after this whole vaccination, and a lot of people were suspecting maybe they're related. But then again, does all these antibody, are they trying to attack your heart in the meantime, and are they actually working on your pacemaker cells, just the way that I mentioned? Maybe could be one of hypotheses. Any add-on?
I think I probably just can add some comments. I think it's a great question. I mean, in terms of COVID-19 vaccine, I mean, it's still debating. I mean, it's good or not. I mean, from sort of scientists in the field of drug discovery development, I think if there is no pandemic, absolutely those vaccines should go through more clinical trials. It's too early to get into the clinical. But at the population level and this pandemic time, so it makes sense to try those But another comment I would like to make, well, even you see a lot of young, folks die, but that if you compare to the old people, it's not fair to say that young people are easily to get die. Still more old people died because of those vaccine and also the COVID-19. So in terms of how to solve it, I guess currently no answer. Still, I think I know there are many research groups to try to collect the data still, to see actually what's actually the consequence of the COVID-19 vaccine to the whole population. So.
For the interest of time, we may have one more question from the audience.
Yeah, I'm working for the COVID vaccine company. So I don't want to make any conclusion, but I want to give comments. So for the mRNA vaccine, COVID vaccine, or protein-based COVID vaccine, so in the label, we all observed myocarditis and the pericarditis happened in the young people, in young men. It's not women. So as the company, we cannot explain that. Why in young men? Typically in
that there was a study showing that there's an increase of ACE inhibitor, ACE receptor something in the males, right?
COVID virus, they always use the AC receptors, but the AC receptors is everywhere. It's in the vessels, on the heart, even in the brain. So, but why only young people? We have this age, 16 to 26 age. So young men, they get the myocarditis and pericarditis. Maybe later that will be a topic, because for COVID vaccine, main I think adverse events, serious is myocarditis, pericarditis. The other things, I think it's just mild. Yeah, that's my comments.
Very good. Is there any clinical study?
I don't think so. I think now COVID vaccine company is very tight for the funding.
Okay, they might get.
Thank you all. Thank you very much for the insightful thoughts sharing and knowledge sharing. And thank you very much for the active discussion in the discussion section. And we've covered a fascinating spectrum today, enabled the prediction and clinical translation using AI tool. And let's give our speakers another round of applause. And also please stay tuned for our next session.